Hi, Paul. How are you? Hi, Terry. Hi, Iris. How are you? Yeah, I'm just setting up now on my YouTube channel. I'm just getting it all set. I've not actually planned anything. I'm editing my video because I just finished my group session. I'm good, thank you. Yeah, I'm good. I'm just setting up on my other channel. Hi, Tony. How are you? Excuse me. Oh, that's good. That is good. Hi, if you are joining, there we go. You may have answered this, but do we need to know about, that's right, Mara. No, you don't need to know. If they're going to ask a question on that, they will literally give you information about it. But no, you don't need to know that. Right, where do I want to view? Over there. And yeah. Guys, if you have any questions, please do. Ask away. I am just setting up on my channel to answer. I didn't do a live last night. I did a live obviously on Sunday night, but I didn't do one last night because I wasn't feeling too good. I had a bit of an ear infection. And so I was feeling a bit drained. So I thought I would wait, but I've just finished my group tuition session tonight. I'm editing my videos right now, so I thought I would jump on here to answer any questions that people might have. So if you do have a question, please do ask away. And yeah, I am happy to answer any questions that you have. If not, then what I can do is just start doing some questions that we covered in previous sessions. So I am pretty easy to do whichever one you want to do. So yeah. Right, so let's have a look. I don't actually remember what last paper was it we're doing. As a year 12, to cover all of the AS content, when should I start revising for mocks in Gin? You should already be revising, really, Elodie. What I would suggest you do is make sure that you are covering the fundamental parts that you require for your exam. So it's one of those ones where the more you can do now, the easier it will become. The longer you leave it, the harder it will become. So yeah, absolutely, you should be doing now little and often. My exam for biology paper one is on the 12th of May. So you've still got, what are we on, the 29th, 30th? So you've still got 11, 13 days to go, Sarah. You can still do this. You have the time. It's about whether you put the effort in to do it now, which is the hard part. It's so hard when the exams are upon you, but make sure you're revising the stuff that you hate and focus on that. Do you need to know the specific chemicals used in plants, chemical defenses? No, you don't. You need to know. So with regards to that one, you do need to know the some examples, but if they're going to ask a question, it'll be more about the information they've given this time in the question. So response, if I just put the specification up then, give me a second. So they specifically said in this one, so if I jump across here, so plant defenses against pathogens, production of the chemicals, but they are not going to want specific examples. And then with regards to the limiting the spread. They've actually put it down to just being the caloes. And when it came to the herbivory side of things, they took that out as well. On module 5, they gave you much less on the plant response defense to herbivory. So where is it? Yeah, so it's the type of responses, plant response to abiotic stress and herbivory, but they have said that that one specifically, and again, I'll put that on my slide here, it is alkaloids and pheromones only on that one. So they're the only things that they've actually given you. Chemistry's tomorrow and biology next week. I'm petrified. Oh, I'm sorry that you're petrified, but remember it's going to be challenging. It's meant to be challenging. And if you access it in the right way, then you should be able to do what you need to do in the exam. But it's hard, and good luck. It is hard. One of the best things to do, though, is to walk in and say, it's going to be hard. I'm going to try my best, take my time, and actually write down stuff, even if you think it sounds silly, because what you might find is the stuff that you write down is going to get you marking points because there's little tidbits in there that you've got which are actually correct. You just can't formulate the bigger picture yet. So try your best. Best way to revise for your mocks that cover the AS part of the A-level course. The best way to revise, I would say, is AIQ chat. And doing the multiple choice questions specifically on that topic area, because that's obviously going to plug what I do. The other thing I would say is do as many past paper questions as you can. And once you do the past paper questions, what you're going to find is that you will get better at answering as the examiners want you to. Can you go from a U to a C? Absolutely. It just depends what it is you actually are doing. Oh, it's not available in Zoom. That's interesting. That's weird. Why is that not available? Maybe I've hit a button that's made it unavailable. For AQA, I mean, if it's AQA or if it is OCR, if it's EdXL or if it is the IB, it doesn't matter. What's important is the exam board asks things in a very specific way, but the content's still the same. If you have the fundamentals of the content, then you will be able to apply it to whatever question is given to you. So it is about getting those building blocks that you have the key parts and then going from there onwards. For the buffer solution question, I'm just going to draw out the hydrogen dissociating and hope for the best. The best thing to do. Iris, are you having problems with the Zoom as well? Can you write something in the chat? I have to say, it's a funny one. When we talk about chemistry, I was doing a session with a student earlier today, and they're doing A-level chemistry as well. And I don't teach A-level chemistry. I don't have an A-level in chemistry, although I've got a degree in biology and I've taught chemistry up to GCSE level. I don't have an A-level in it. And sometimes you can go into more detail than is necessary when you do A-level chemistry for your biology exam. And sometimes you can bring your biology into your chemistry and not go into enough detail. And that's hard. I mean, buffer solutions are all about buffering the pH so that you are keeping it at a controlled level. And so therefore, if you do that, you are going to not affect, for example, the enzymes that are on there, that are working inside there. What's the code? Here, hold on 1 second. Hold on, that did not work. Give me a second. There you go. Oh, maybe I hit a button. Yeah, it says that it's been turned off by the host. I'm probably hit a button. What have I done? I don't know what I've done tonight. Hold on 1 second. Right. Give me a second. Let me stop sharing. I'll see if I can fix it. Oh, let's go back to share. Try it now. If you try it now, it should work. I'm hoping on the chat on the Zoom. Yeah, that's one of the hardest parts as well. Oh, I see some chats coming through. Yay, there we go. There we go. I don't know what I did. I obviously clicked the button to stop it. Freya, once you start revising, you should always start as soon as you get going with biology because there's so much content. Bidonic acids are bases of a salt that's a species. Hi, I am good, thank you. Oh, Freya, so you're doing GCSE, so you definitely need to be doing your revision now and as much as you can possibly do. So yeah, it's a difficult one because whenever you are doing any type of exam questions, you need to know what they're going to ask. And so therefore you need to be able to apply that content any which way you can. So Iris, I'm not sure if you have specific questions you want me to cover since you're in the Zoom. We've done the naked mole rat side of things, but we haven't done this one. Should we do this question unless you have done it? Thank you, Rio. I appreciate that there are companies that do the same thing for £20. I would say, have they got 20 years experience? Are they Cambridge graduates? Are they examiners? Potentially they are. And if they are and they're given the same quality, then absolutely. If you are looking for that, then great. But thank you for joining me tonight and for advertising the other sessions. That is the key thing, is that everybody has their budget that they can go and do. I mean, this actually is £25 an hour of a session, which is the cheapest I can get it for all the resources that I do beforehand, all the planning, all the copying that you get of my resources, my diagrams, etc. So I understand it's not for everyone, and that's fine, but thank you for joining me. Thank you, Paul. That's really helpful. Right, let's do this question. So I actually do want to do a bit of teaching tonight because I'm only going to be on for about half an hour. So if we are looking at this question, then we're told that the ice fish live in very cold water. We're told that they contain biological molecules that allow them to tolerate cold temperatures. So straight away, they're giving you little hints in the question about it being cold. Now, they're expecting you to be able to know the difference between whether it is anatomical, whether it is physiological. So when you're looking at these types of questions, you should be saying to yourself, what are my key words? What are my keywords? What should I be writing as best I can to answer this type of question? So in this case, we are looking at whether or not it's anatomical. I'm trying to use those words too quickly. Is it physiological? What is it that's actually being changed? So the adaptations can be grouped into three. Which category is a cold tolerant molecule? So the cold tolerant molecule is obviously to do with the biochemistry. So therefore we would say that it is the physiological. It's not the anatomical one. So we can say it's not that one. Sorry, I've got a really itchy ear because I've got a bit of an ear infection. So excuse me if I scratch my ear. Okay, moving on to this next one then. So one example of a cold tolerant molecule present in ice fish is a modified form of the protease. So again, they told us it's a protease. It's an A, so it's an enzyme, which they actually then go on to say it's an enzyme, and they called it the trypsin, obviously. It's a protease because it breaks down proteins. So then we look at this one, and it says, okay, we've got trypsinogen, and that trypsinogen is going to be converted into trypsin, and it's converted into trypsin because we have this calcium ion binding site. So therefore, calcium is going to bind to it. Now, we know that calcium is an inorganic ion. So we know that the calcium is actually a cofactor because what we've done is we have activated the trypsin, the enzyme, to have the correct active site to be able to break down the proteins. Now, trypsinogen is an inactive form. It is what we would call a precursor. So the fact that it's a precursor, the fact that it's an inactive form, means that it's not going to be working. So what we need to understand is that this conversion from this inactive form into the active form is actually carried out by another enzyme. So here we can see we've got an enzyme, and that enzyme is going to be immobilized in the sense that it is actually attached to the wall of the small intestine. So that's a way in which we naturally immobilize enzymes, in that we put them into the wall of the small intestine. And what that enzyme is going to do is that enzyme is going to convert the inactive form into the active form by adding the calcium ion on. So although this is not what the question is asking me, I'm going to add this in here. We have got the inactive precursor. That inactive precursor is a protein. That protein was made through transcription and translation. What we are then doing is we are modifying that protein. So we are doing what's known as post-translational modification. Whenever we have a precursor and we're going to activate it by adding something onto it, for example, this case, we're adding on the calcium ions, we are activating it. So therefore, we are doing a post-translational modification. And that is one of the ways in which we can talk about gene regulation. By having it as a precursor, by having it as an inactive form, means it will only be activated in the right conditions when there's lots of calcium ions present. So that would definitely be something that you would want to link it to. Now, the other thing to note is the calcium ion is binding to a specific site. And so because the calcium ion is binding to a specific site, and that site is away from the active site, so obviously this section here is the active site. So because it's away from the active site, that's therefore saying it's acting like a non-competitive inhibitor. So it's important when you're going through these questions that you're looking at different ways that they could ask questions on the topic area. So now let's have a look and see what it says. Calcium ions bind to lots of different things, whether it's troponin or whether in this case it is the trypsinogen. They do like to bind to lots of things. I swear, ever since you've come on, I used to get so stressed out when you were in. And now I realize that actually lots of people will always try and throw teachers off, and it's about what you do with it. Binding to the allosteric site. So this is an interesting one because you're saying binding to the allosteric site, but in this case, in the question, they've told you it's a calcium ion binding site. So you could use the term allosteric site if we're looking at the non-competitive inhibitor, but what we want to do is we want to try and use the information that's there without tripping ourselves up by saying that it's an allosteric site. Although we use that term for a site away from the active site, what you want to use is the information they've given you in the question to be sure that that's what they'll be looking for. Because what they could say is that actually you are only using terminology that you've memorized. So you want to refer it back to the question whenever you are looking at it. But yeah, it would actually bind to an allosteric site. You could refer to them, yes. But we, again, so Mrs. Lana Ray, if that's the name that you want to have, that would be where you're bringing in more of your chemistry knowledge than you are for the biology side of things. We wouldn't go into that much detail or use that terminology, but you could use it in the exam, but you need to sometimes be careful. You don't put in the bits that won't get you the marking points, which is really hard. It's especially hard when you're doing biology and chemistry. It's like, which one should I use? So you won't be penalized as long as the information that you give is correct. But what I would say is just be a bit hesitant that you don't go into too much detail that takes you off on a tangent that loses you the marking points. Okay, so looking at this one then, what have they told us? So we've got a group of students investigating the effect of temperature. So straight away, I want to say to myself, well, this is my IV, my independent variable. On the activity of two forms of trypsin. So I've got two forms. I've got human trypsin and icefish trypsin. So technically, I've actually got two IVs because I've got the type, the form of the trypsin, as well as the effective temperature on it. Oh, good luck for your mock tomorrow. All you can do is try your best. That's the key thing about it. So what are we going to use? We're going to use 10 centimeters cubed. So they've given me my volume. We're going to use 5% trypsin solution. So they've told me my concentration. We're going to measure it at 10, 20, 30, and 50 degrees C. So they have given me 5 values for my independent variable. So that's important when doing this. Yes. Yes, I can normally work out the questions for paper 3 based on what they've asked in paper 1 and paper 2. Because if they've asked a specific question in paper 1, like they did a lot on, let's say, respiration and photosynthesis in paper 1, then that tells me that they may mention respiration in paper 3, but they won't go into as much detail about that. So it's about how you look at it. Now, if you're mocks, so if you are doing, I'm assuming if you're doing paper, if you're talking paper 3s, you're obviously A level, so you won't be doing the 2023 as mock, or you've done 2024 as your mocks. So if I say to you that the paper last year, they did a lot on respiration in paper 3, more photosynthesis in paper 1, but they linked respiration into paper 1, but went into detail, but only specific detail in paper 3 on one section of it. So it's a fascinating way that they can ask it. So I can make a prediction normally in the topic area, but I can't know it all, is what I would say. But after paper 1, I will ask students to tell me what they've been asked in paper 1 so that I can then formulate what I think paper 2 or paper 3 is going to give, because obviously module 1 and module 2 get asked in all 3 papers. So if they don't ask about DNA replication in module 1, in paper 1, then that would tell me that potentially DNA replication might come up in paper 2 or paper 3. So it's important to know what comes up in each of the papers. You'll see an overlap of it. So yeah, you can normally work it out, but you can't guarantee that you'll know how to do it. Okay, back to this one then. So we carry out each trial in the same buffer solution. Guys, I have no idea if you're asking me questions on the actual live. Give me a second. There we go. No, I don't think we are. It's not working there. Okay, so if we then look at this one, I'm hitting too many buttons here. Each trial carried out with the same buffer solution. So the same buffer solution. So we've got a control variable there. we are controlling the pH. You're welcome, Lily. Measure the enzyme activity by recording the area of the gelatin on photographic film, and that is broken down over a set time period. Then you're going to calculate the rate of the enzyme activity at each of the temperatures. So when you look at a question like this and they're asking you, so this is a module 1 question. And the reason I know it's module 1 is because it's asking me about improvements to a practical. So when it's asking me about improvements to a practical, I know that that's to do with module 1, because that's about my practical experience and how I can apply that practical experience to the situation. So 2 improvements that would increase the validity of the student's investigation. That's the important part, making sure that you know how to increase the validity. Hi, welcome. So things like they've done control variables, but have they controlled all of the control variables that are possible to control here? So have they controlled every single bit of it? So if it's not mentioned in the text, is there something else that I can talk about? Well, they said to use the same pH buffer as a control variable. So is that pH buffer, are they talking about the concentration? Or do we need to think about the volume? Have they mentioned the volume of the pH buffer? No, they haven't. So I could say that doing that is going to make a difference. Now, they've said that they are going to record the area of the gelatin on the photographic film that is broken down. Well, if I have a gelatin block and I don't actually control the thickness, i.e., look at my surface area to volume ratio, is that going to have an effect on this? How much I cost per hour? If you're looking for for example, this with regards to my group tuition sessions, they are the ones that I do at a £25 an hour for this, but that's not just £25 for the hour you get with me, but then you've also got all the planning that I do before and the resources afterwards. So you get a copy of the recordings and all my notes and diagrams that they do. With GCSE, it really depends whether I have the time to be able to do it, unfortunately. For GCSE, I do more A level than the GCSE now, because with GCSE, it's not as big a need or an ask on that one. Why are you watching this when you have your exam tomorrow? Well, because you just want to have a break, or because you think you might get some information. You're not sure why you ended up here, but you're interested. Thank you. That's what I want to do. I want to make sure that everything that I give you is information that's going to help you if you are studying for your OCR A level. But if you aren't, if you're actually studying other exam boards, some of these skills actually go across. I've taught International Baccalaureate, I've taught I teach EdXL and AQA as well. And therefore, these skills that I'm teaching you will help moving forward, because they're practical skills that you could use in a degree, potentially, if you go on to do some biological degree. Anyway, back to this. So I diverge. What else have we got? Well, it's interesting because here, looking at this one, so they obviously did 5 values of their independent variable, and they have 10 degrees C intervals. Well, if they have 10 degrees C intervals, if we're talking about enzymes and we're talking about trying to find the most effective activity, so we're looking at that optimum temperature, how can we work out that optimum temperature with 10-degree intervals? So what we might want to do as an improvement is actually have 2 degrees C intervals or 5 degrees C intervals. So it just depends what you want to give them. They've said, what would the improvement be? And then why? Explanation as to how that would increase the validity. If we have intervals of the temperature which are closer together, we're going to be able to more accurately estimate that optimum temperature. Because if we're looking at our graph and we can see we're increasing and then it drops down, at what point here is the optimum if I've only tested at, let's say, 30 and 40 degrees C? Am I absolutely certain that it's 35 degrees C is my optimum? Or am I actually not sure and therefore I should do 30, 32, 34 and so forth? So it's ensuring that you want to give the information that will help you. And you, Paul, you're right. They haven't mentioned anything about timing. They said over a set time period. What is that? Is that 5 minutes? Is that 1 hour? Is that 24 hours? You need to be careful that you give realistic information when you do that. Would you have to say intermediate temperatures or interval temperatures? I would say interval temperatures, but actually it doesn't matter which way around. If you're talking about intermediate values in between, then that would be enough to show that you're saying those numbers in between. So intermediate temperatures, the ones in between, the 30 to 40, or interval temperatures of 2 degrees going up. So it really just depends what you want to say to answer that question. But the key thing is different students, just to be aware, there are 20-ish thousand students that will sit this exam. Now that's talking about the A level, not the AS, but 22,000 students sat it last year. Of those 22,000 students, you're never all going to write it exactly the same way. So what we do as examiners is we look at your answer, and as long as you haven't given it as being too basic an answer, and you have given a description which is accurate, we can then give you the marking points. So don't get hung up on wording to say that, well, I wrote this, but actually I needed to write that, unless it is key terms. If it's key terms, like, for example, talking about restriction endonucleases, those are the types of words you need to know. Photophosphorylation, for example, there's specific wording that you need to know, coenzymes, etc. But when it comes to things like intermediate temperatures or interval temperatures, then actually they can be used interchangeably, and so that's not an issue. And I say that because I actually stressed out in my A-level exams equivalent in the fact that I didn't think that I would get the marks if I didn't write it a very specific way. And you'll hear me, I fumble with my words sometimes, and I try to be very specific in what I'm saying to help you with those key words. But I speak too quickly. I sometimes write too quickly, and then it comes out as a troll on the paper. You can see it as I'm doing it here. Because of the way my brain works, I do things slightly differently, and therefore I now know that I have to slow myself down. But I also know that if I try and write too much, my muscles get tired. I don't manage to get it down. So I've learned to write in bullet points, and therefore I'm specific, I'm logical, and I write it in bullet points so I can get the marking points. So it doesn't have to be an essay. Even though if you're doing AQA, it does have to be an essay. With OCR, it absolutely doesn't. Your biology exam is on Thursday, and then you're finished, as in you've got no more worrying to do, but still content to be taught, or you're actually done. Chidima, that's an interesting, weird feeling, because you'll have a break and then be like, what do I do with myself now? Start revising, start getting ahead. Okay, let's look at this question. Appropriate units to represent the rate of enzyme. So we know it's about a time period, so that's important. We know it's a time period, and therefore we know that we're going to be talking about the area. So because we know it's the area, we either need to do millimeters squared or centimeters squared, because we're looking at area. And because we know it's got time, we therefore would want to have that in either seconds or minutes. So you can have millimeters squared per second, or centimeters squared per second, or millimeters squared per minute, centimeter squared per minute. So it is important that you note that rate should always be quantity over a specific time. So, and it is important that you write it in the right way. Oh, wow. Third year medical student here. Wow, well done. It's one of those things that getting in is the starting point, but then it absolutely then gets more and more complicated as you go. You are ready, Chidama. You are ready. You've actually got this, okay? Do you do these sessions weekly? Yes, I do. I normally do it on a Monday night, but I wasn't feeling very well last night. So I jumped on here tonight after I did my group tuition session. And I also will do one tomorrow night. Wednesday is my normal night that I definitely do it. So I'll be on about 8:30 tomorrow night to do it. Is A-level biology not just chemistry? It's an interesting one. It's not just chemistry because I never did an A-level in chemistry, but I still managed to get my biology degree. I had to do chemistry modules in my degree, which was hard when I hadn't in Scotland. We didn't have to do all three. I chose biology and that was all I did. Whereas now you have to do it all three, which is the right thing to do. Do you do National 5 biology? I don't, Kirsty. Although I haven't, I mean, I've looked at the specification. It's not that complicated, but I haven't looked at the actual exam questions. That's the different part. Different exam boards ask things in different ways. And so I'd have to look at the questions and the past papers to figure out how they're going to assess you. But with any of the exam boards, if you are a teacher who knows what to look for, and I did examination for a number of years because it was good CPD to begin with. It gave me a bit of money, but actually the CPD I got from doing that, as in the professional development that helped me, helped my teaching. So any teacher who's done any type of examining could potentially teach you it. It's just about making sure they know your course and are specific to what you need to do. Your brain isn't braining. To be honest, Tony, half the time my brain isn't braining either. They do help each other. I have to say, I'm now with the chemistry, I'm reviewing the A-level chemistry content. And the more I do that, the more it makes the biology make sense, which is bizarre. I've been teaching this for over 20 years, and yet there's still parts of it I forget. There's still parts of it that I have to go over again and again and again. And so therefore, by doing this, by going back over the other content and figuring that out, helps me to understand it, to then teach it better. And so like today I was talking to a student who does chemistry and biology, and we were talking about the fact that ATP, when it's hydrolyzed, releases energy. And so it's a difficult one because obviously when you're doing ATP and you're looking at the ATP and you talk about breaking the bonds, when you break the bonds to release the inorganic phosphate, so breaking it down to ADP, energy is released. And he was like, but if you break bonds, then you need energy. So how is that? How does that work? And so we were talking about the fact that if you understand that you have energy needed to break the bonds and energy needed to make the bonds, then what you have is that the water that goes in to hydrolyze the ATP down into ADP and the inorganic phosphate is breaking and reforming bonds. But what you also have to understand is the ATP, actually, you have phosphate close to another phosphate. Well, that third phosphate is actually being repelled because they are negatively charged particles, that they actually repel each other slightly. So there's a strain on the molecule anyway. And therefore, when that inorganic phosphate, the third phosphate, is broken off, what actually happens is that results in a product being made, ADP and inorganic phosphate, which is more stable and has less energy. And so therefore, that's the form that it stays in. So we need to have the energy put in to join the bonds back together to make it a ATP, the adenosine triphosphate. And when we hydrolyze it, energy is actually released because of the fact that those products are a lower energy and also more stable. So that's where the chemistry part needs to come back in when you're doing this. It depends when I can. So it's either 8:30 or 9 o'clock, but tonight was a bit later because I was editing my video from my group tuition sessions. Would I recommend, Stephanie, are you year 12 or year 13? So with regards to if you're year 13, I would recommend if you are making your own flashcards, only do it on the topic areas that you hate. If you're buying them, make sure you're actually using them in the right way so that you are then testing yourself and doing active recall rather than just passively going through them. Iris, to answer your question, would we format the rate in a fraction or a single line? Format the if you're year 12, Stephanie, I would say start doing them yourself. Because you've got the time. Do it now, do it over the summer, get all the flashcards for the previous year 12 content sorted before moving into year 13, and that will consolidate that knowledge. So when you say, Iris, can you explain to me what you mean by would you format the rate in a fraction or a single line? What do you mean by that? Sorry, my brain's a bit slow because I am at the end of the day. Oh, it's now changed color because it is telling me it's time for my bed. Let me just quickly check on these. I had to do tons of biochemistry in first year of degree. Yeah, it's an interesting one. I had to do some, but not loads. And it was hard. It is hard. Yes, if you've got a 6, you'll be absolutely fine in A-level biology. Okay, so it's important you are going to either write millimeters squared per second or millimeters squared per second minus 1. Don't do a mix of the two because you will lose your marking point on that one, Iris. Yeah, Omar, if you're getting a grade 6, you'll be absolutely fine doing A-level biology. As long as you're getting a 6/6 if you're doing combined or a 6 if you're doing biology on its own. You can also do A-level biology with a 5. You just have to work a bit harder to pick up the knowledge that you're missing. How much should I use to make biology postcards? It can be. It is so time consuming. Right now, I'd say it's better actually if you're year 13 to do past paper questions rather than doing the flashcards. You're doing combined. Yeah, so if you're getting a 6, a 6/6, you'll be absolutely fine in doing A-level. I am actually just going to finish, guys. I am going to go to my bed. So although you are just entering, I'm literally going to finish. And I will say good night. I will be back on tomorrow night, as usual. After I've had my clients earlier on, I will be back. And so I will do more tomorrow. So if you have specific questions, please do send me a message on my YouTube channel, on the video, or on here, because that will then allow me to respond. It depends on the Wednesday of whether I'm going to go 8:30 or 9. It depends on what I'm doing. I should be able to do 8:30 tomorrow night. Make sure you're capable of self-control. Start revising from the beginning. Absolutely. It is hard. Yeah, do as much as you can, and that will help. But guys, thank you for joining me, and I will see you tomorrow.